Okay, welcome to the Autumn 2 Week 5 exam video on osmosis. So, the first question asks us to draw a ring around the correct answer so we can finish each sentence. So, first one's nice and straightforward. Water moves into cells and out of cells by osmosis. So, osmosis is only for water, and so the other two options, active transport and diffusion, completely incorrect. The water moves through a semi-permeable membrane. Sometimes we also say a partially permeable membrane, but the only one that makes sense here is a semi-permeable membrane. So you get 1 mark for each of those. Okay, the next part of the question is going to talk about some strawberries. So, some sliced strawberries are sprinkled with sugar. After 2 hours, the slices of strawberry have shrunk a little. Now, I underline that because that tells us if the strawberries have shrunk a little, it tells us that their mass has gone down. Their mass has decreased and the sugar has formed sweet tasting syrup. So, let's have a look at the picture. Right, we've got the strawberries with the sugar sprinkled on them and then the strawberries 2 hours later. So, the next question is going to ask us to explain why this has happened. So, why have the strawberries shrunk a little? And why has sugar formed a sweet tasting syrup? But mainly, why have the strawberries shrunk a little? So if you look at what I've done to the diagram, you've got 2 parts here. You've got the inside of the strawberries, and then you've got the outside where the sugar is. Now, sugar on the outside of strawberries, that's going to be a really concentrated solution. Very concentrated. Very little water on the outside of strawberries. On the inside of the strawberries, you're going to have still some water, and so that's going to be the more dilute part. So I've labeled it just like we've labeled all of our osmosis questions in the past. So D for the dilute part inside the strawberries, C for the concentrated part, which is the sugar on top, and then an arrow going from D, the dilute part, to the concentrated part. And that tells us why the strawberries are going to shrink, because the water they have inside is going to move out of them and is going to combine with the sugar, and it's going to dissolve the sugar, and that's why we get a sweet tasting syrup. So let's see how we get the full 4 marks for this question. So the first thing we've got to do is point out the differences. So we've got a more concentrated solution outside the strawberries, so for mark number 1. Mark number 2, it's more dilute inside the strawberries. Then mark number 3, water is going to move out of the strawberries by osmosis, so we must name that process. And then finally, to get your 4th mark, you have to have mentioned that it's going through a partially permeable membrane, or you could have said, instead of partially, you could have said a semi-permeable membrane. So to get the 4 marks, we need to mention all of those things. Okay, on to question number 2. So, some pupils set up this practical to investigate osmosis. So they filled 2 pieces of semi-permeable visquing tubing with different liquids and left them both in a beaker of 15% glucose solution for an hour. So if we have a look at the diagram, so the solution here is 15% glucose. Now what you should keep in mind is more glucose means less water, so more glucose actually means more concentrated. However, the 2 tubes have different kinds of solutions in them. So this one, let's start looking at this one, the 10% glucose solution. So 10% glucose, that means more water. So that's why I've labeled that tube with a D, because it's dilute. And then with reference to this tube, the 15% glucose solution is more concentrated, because 15 is higher, which means more sugar, less water, so more concentrated. So, for this tube, because it's more dilute inside and more concentrated outside, the water is going to move out of the tube and it will get smaller. Now for this tube, it's a 20% glucose solution. So that means there's actually less water inside than outside. So for this tube, the outside is dilute, the inside is concentrated, so water is going to move into that tube. And so that's why you can see that this tube looks like it's shrunk, this tube looks like it's gotten bigger. So The question asks you to describe and explain the likely result after 1 hour. So, for tube A, describing the results, water is going to move out of the tube, and then our explanation, because the solution inside is more dilute than the solution outside. You could phrase that differently. You could say that the solution inside is dilute and the solution outside is concentrated, but as long as we've got this key idea that inside is more dilute than outside. Then for tube B, water is going to move into the tube, and that's because the solution inside is more concentrated than the solution outside. So 4 points, need each of them to get the 4 marks. Okay, question 3 is a pretty typical question on the osmosis required practical. So the GCSE pupil carried out a practical investigating how does the concentration of glucose solution affect the percentage change in mass of potato cylinders. A pupil investigated the effect of different concentrations of salt solution on chip mass. So he recorded the mass of each of 5 chips. He placed each chip into a different concentration of salt solution. After 1 hour he removed the chips, then recorded their masses once more. Now one thing we've got to be careful of is it's referring to chips here and then potato cylinders up here, so they mean the same thing. Potato cylinders, chips in this question are the same idea. So let's have a look at the results. Okay. So we can see we've got 5 different concentrations. Original mass, that's the mass of the potato cylinders at the beginning. Final mass, that's their mass at the end. And then we've got the percentage change in mass calculated. Now, the formula for percentage change in mass you looked at on your knowledge questions on osmosis. So to find percentage change, you take the final mass, subtract the original mass, and divide by the original mass. Now lots of people make the mistake here and divide by the final mass, but you divide by the original mass. And then to get a percentage, we multiply by 100. So the first thing we're asked to do with these results is actually to find the value of x in the table. So we want to find the percentage change in mass for the 0.4 mole solution. So we take the final mass, 2.3, we subtract the original mass, 2.5, and we divide by the original mass, 2.5, and when we multiply that by 100 we get -8, and the minus here is really important because percentage change in mass can be negative, so it's got to be -8. If you just put 8, you only got 1 mark. Okay, now the next part of the question, so suggest why the pupil calculated the percentage change in mass as opposed to just change in mass in grams. And there's just one simple reason for this. When we have a percentage change in mass, we can compare our results more easily. So it's just easier to see the differences when we have a percentage change in mass. Okay, the next part. Identify the concentration of salt solution that caused the chip to gain mass. So the key thing here is gain mass. And if we go back up to our table, there is only one solution where the chip had a positive percentage change, and it's this top one here. So the original mass was 2.7,